Welcome to the BioInsights, where science comes alive.
Have you ever wondered how doctors make those split-second, life-saving decisions when they're faced with a totally unknown infection? Today, we're acting as medical detectives. We're going to uncover why the tiny, microscopic structural differences between bacteria are, quite literally, a matter of life and death. Figuring out the kind of armor these culprits wear, that is exactly how we crack the case.
To solve this microscopic mystery, we're going to follow these 6 steps. First, the bacterial detective story, followed by the Gram stain procedure, a tale of 2 walls, toxins and key differences, the high-stakes medical reality, and finally, identifying the microbial suspects. All right, part 1, the bacterial detective story.
Imagine you're a detective in a hospital dealing with an unknown infection. A patient is incredibly sick, you have multiple bacterial suspects, and you need a quick clue right now before it's too late. So why does knowing if it's gram-positive or gram-negative actually matter in a high-stress place like the ICU? Well, normally, when you draw blood or urine culture, it can take days to fully grow and identify the exact species of bacteria. But here's the thing, a patient in the ICU doesn't have days. By immediately categorizing the bacteria into one of 2 broad groups, gram-positive or gram-negative, doctors get this instant, vital clue. It completely dictates which life-saving antibiotics to use before those long-term culture results even come back.
Moving right along to part 2, the Gram stain procedure.
The primary interrogation tool we use on these suspects is an absolutely ingenious method invented by a scientist named Hans Christian Gram way back in 1883, and it is still a cornerstone of medicine today. We use a super precise, 4-step chemical sequence to force the bacteria to reveal their true structural identity. Step 1. We flood our microscopic suspects with crystal violet. It's a dark purple dye that gets sucked right into the cells. Step 2. We add iodine. You can think of iodine as a mordant, basically a trap that locks that purple dye inside. Third, and this is the critical one, an alcohol wash. The alcohol acts as a decolorizer, actively trying to wash that purple dye right back out. Finally, step 4, we add a red or pink counterstain called safranin. How the bacteria react to this exact chemical interrogation, that gives away their entire identity. And here is your golden rule. If you take away one thing, remember this. P for positive, P for purple. Because of their structure, gram-positive bacteria trap that initial crystal violet dye so tightly that the alcohol wash cannot budge it. They stay a brilliant purple or blue under the microscope. Meanwhile, our gram-negative suspects completely lose that initial purple dye during the alcohol wash. They basically become transparent. So they end up soaking up that secondary safranin dye, showing up as a bright red or pink.
Okay, part 3, a tale of 2 walls. To understand why they stain so differently, we really have to zoom in closely and examine the physical armor protecting these microbial suspects. It all comes down to a protective shell wrapped around their inner plasma membrane. This armor is the cell wall, which is largely made up of a complex polymer of sugars and amino acids called peptidoglycan. And look at this massive discrepancy. Gram-positive bacteria have a remarkably thick wall. We're talking anywhere from 20 up to 80% peptidoglycan. Gram-negative bacteria, on the other hand, they have a super thin layer of it, maxing out at around 15%, but usually hovering much lower than that.
So, what does this actually mean for the bacteria? Well, the gram-positive wall acts like a thick, rigid brick wall. It is incredibly robust, measuring roughly 25 to 30 nanometers thick. And actually, this is exactly why it traps that purple dye so well. Gram-negative bacteria, however, have a wall only 10 to 15 nanometers thick. But don't count them out, because they compensate for this thinness with a totally sneaky secondary defense, an entirely separate outer membrane that is highly elastic and incredibly rich in slippery, oily lipids.
Which brings us to part 4, toxins and key differences.
Beyond just the physical thickness of their walls, these 2 types of bacteria are packing entirely different chemical weapons. This outer lipid shield, or LPS, is exactly what makes gram-negative bacteria so incredibly dangerous when they get into the bloodstream. Because gram-negative bacteria have that extra outer membrane made of these lipopolysaccharides, they inherently carry what we call an endotoxin. It's literally built right into their skin. When this LPS enters the human body, it is highly immunogenic, meaning it can violently and dangerously trigger the human immune system.
Conversely, gram-positive bacteria completely lack this outer LPS membrane. But they aren't defenseless. They contain a really unique substance called teichoic acid woven directly into their thick, purple-staining outer armor. Now, while it does act as an antigen that our immune system recognizes, it is typically not as violently immunogenic as those endotoxins found in the gram-negative bacteria.
If we put these profiles side by side in a sort of cheat sheet, it reveals some really stark physical differences. Beyond the outer membrane being absent in gram-positive and present in gram-negative, just look at the other adaptations. Gram-positive bacteria have highly developed mesosomes, while gram-negatives are poorly developed. If they have tails or flagella for movement, gram-positives only need 2 structural rings to anchor them in their single thick wall. Gram-negatives, they need 4 rings to anchor through that complex double membrane system. And crucially, gram-positives generally release exotoxins, while gram-negatives carry those super dangerous built-in endotoxins.
All right, part 5, the high-stakes medical reality. Let's see how these microscopic details actually dictate hospital survival rates. As the quote says, gram-negative bacteria are tougher because their outer membrane acts as a shield. This makes them more resistant to many antibiotics and harder to treat. Gram-negative bacteria essentially use their oily outer membrane as an impenetrable shield against many common medications. Remember that alcohol wash we talked about earlier? Well, just like it washed away the dye, that oily, lipid-rich outer membrane on gram-negative bacteria actively repels many of the drugs we try to throw at them. It makes them notoriously difficult to treat.
Take a classic antibiotic like penicillin, right? Penicillin works by specifically targeting and destroying the peptoglycan cell wall. Because gram-positive bacteria have their massive peptoglycan wall just fully exposed out there, penicillin acts like a wrecking ball, making them highly vulnerable. But for gram-negative bacteria, that sneaky outer LPS membrane completely blocks the penicillin from ever reaching the thin peptoglycan layer hidden underneath. Furthermore, treating gram-negative infections is a terrifyingly delicate balancing act. If you use a strong drug to forcefully blow apart a gram-negative bacterium, you risk a massive, sudden release of those LPS endotoxins into the bloodstream. That violent release can trigger a massive immune system overreaction, a deadly condition known as sepsis.
Finally, part 6, identifying the microbial suspects.
Now that we deeply understand their weapons, their armor, and their weaknesses, let's officially unmask the most notorious bacterial suspects in each category that doctors face every single day. Here is our lineup of gram-positive purple-staining culprits. These are the bacteria with the thick walls. They include heavy hitters like Staphylococcus and Streptococcus, which, is responsible for common ailments like strep throat. But it also includes incredibly dangerous pathogens from the Clostridium family, responsible for severe diseases like tetanus and botulism, as well as the Bacillus family, which actually includes anthrax. And on the other side, we have our gram-negative, pink-staining culprits. Because of those lipid shields, these include highly resistant bacteria that are massive problems in clinical settings. We see things like Escherichia coli or E. coli, a major cause of urinary tract infections. We also see the suspects behind major global diseases: Salmonella causing typhoid, Vibrio causing cholera, and Pseudomonas, which is infamous for causing severe, totally drug-resistant hospital-acquired pneumonias. By simply knowing P is for purple and positive and understanding the thick, rigid walls versus the thin, oily, toxic shields, you now see the battlefield exactly as doctors do. It's like a giant microscopic puzzle, and you've got the key. But it leaves us to wonder, as gram-negative bacteria continually adapt their outer lipid shields, how will modern medicine evolve to overcome this growing crisis of antibiotic resistance? That is the next great case for our medical detectives to crack.